share my screen with you. Are you seeing the worksheet?
Yes.
Yeah.
Okay.
It's very small though.
Okay. Oh, I just made a big mistake.
Professor, did you see my email?
Mostly, no, I didn't.
Oh, okay, that's fine.
So if I have an email from you, after class, I'll look it over and get back to you about it. Okay. Is that bigger?
Yes.
You see it better now? Okay. At the top of this worksheet is a diagram of the cell cycle. We didn't really talk about this in the lecture about mitosis and meiosis, but it's an integral part of cell division. Between the divisions of cells, these various phases occur, G1S and G2, and after G2, then you have mitosis, which we went over in detail. And the description of what happens in each of these phases is given here on the right. So the first question is, what's the difference between G1 and G2? Who would like to address that question?
My name's Katrina Bibbard.
Okay, go ahead.
So what I put was G2 checks the replication of DNA, while G1 gets like the cell ready for replication. So the cell grows and the proteins are made and everything. But then G2 checks replication. So then the DNA was copied correctly. So there's less mutations.
Yeah, that's a good summary of the difference. Let me just expand a little bit on what you said. You remember that one of the purposes of mitosis is to allow the organism to grow, to produce more cells, and if there's an injury, to repair the damage. And for that growth, new enzymes, additional enzymes need to be synthesized and nutrients need to be collected in order for growth to occur. And the point I'd like to emphasize that you made, what I'd like to emphasize about G2 is that that is a checkpoint to check for errors. You remember G2 follows S, and S is for synthesis. So after DNA is synthesized, there's always a chance that copying errors have been made. Well, in G2, there's a check for that. And if there's a copying error, either the cell fixes it with the machinery that is available, or if it's too large an error, the cell will abort. Okay, speaking of S phase, that's the second question. What are the 6 key events that occur with respect to DNA in the S phase? Who would like to describe the 6 steps of DNA synthesis? Anyone want to volunteer? How about Justin Hansen?
Huh?
I don't.
Okay,
I don't know what I'm on right now.
let me help you out.
Okay. So helicase, the SSB, the primase, the DNA polymerase, the RNA, and the DNA ligase.
Yeah, so the reason I make a big deal about this is that the S phase sounds fairly simple, synthesis, but I'd like you to make the connection between the S phase and what actually happens in S. It's these 6 steps of replication. So for each strand of DNA, helicase comes along, separates the strands. Then you have the SSBs, which keep them from going back together momentarily. And then primase is critical for making this little red primer. It's a very short piece of RNA, not DNA. There's an RNA primer here that's absolutely necessary in order for DNA polymerase to work. So DNA polymerase does the actual copying, but before it can start, these other 3 things have to happen. And it starts only where there's a primer. And on the leading strand, it is continuous because remember the copying is always from 3' to 5'. So it starts at one end of a DNA strand and goes all the way to the other end without stopping. The problem that it has is that there are 2 strands. And on the other strand, the orientation is reversed. That is, it's 5' to 3'. DNA polymerase cannot go 5' to 3', so it goes 3' to 5'. But in doing so, it has to wait for helicase to open up the 2 strands. And so the lagging strand is discontinuous, and there has to be, the primase continues to have to lay down little primers every short distance. DNA polymerase has to synthesize, and then RNase H removes the primer, and DNA ligase joins the so-called Okazaki fragments. So this all happens very fast, and it happens during the S phase. The third question is, why would thymine dimers be relevant to G2? G2 follows S.
I'm probably way off. Is the thiamine what's helping to repair the mutation?
You're on the right track in that you have associated thymine dimers with mutation, but they're actually the cause, or can be the cause of the mutation. So if you recall, the thymine dimers cause a kink in the DNA strand. And when DNA polymerase comes along, copying the DNA, comes to a kink, it makes it easy for the polymerase to make a mistake. You put the wrong base in or leave a base out. Anyway, it leaves a mutation. And so in G2, the cell checks to see if that has happened. If a thymine dimer has made a copying error, then it will correct it.
So I was completely backwards. It doesn't
That's
repair
right.
it, it makes it.
Yeah, you were on the right track, but you had the steps reversed.
All right, thank you.
Okay. Then the cell cycle goes around and it comes to this M phase, which spits out, there are 2 cells, one gets spit out, and the other cell immediately goes into interphase for to start another division. So what are the 6 steps that occur in this M phase? Again, in the cell cycle, it looks like it's a short, simple thing, but we all know that it isn't. So who remembers the 6 steps in mitotic, in the mitotic cell division?
My name is Kaylin Helberg, and I think I know. I think it's prophase, prometaphase, metaphase, anaphase. I can't remember the other ones.
Okay, well, you have a very good start.
Through phrase.
Where is it
My
here?
name is Zara, and the last one is through phrase.
Telophase, yeah.
Yeah.
This is a slide that we looked at in our previous lecture, and it has the 6 phases, plus cytokinesis, you remember, is also follows telophase. But yeah, you named them off properly. And what's important that you know is what happens in each of these phases. And when we're talking about the cell cycle, interphase is a very important part of that. And just to let you know, we're going to come back to this in the next slide set, because I'm gonna talk to you about a project that I was once involved in, where we were able to explain the force that moves the chromosomes apart. But that's for a little bit later. Okay, the 5th question is, what's the difference between mitosis and meiosis? And please, someone tell me the major difference between these 2 processes.
My name is Zara, and mitosis produce two diploid, but the meiosis produce a haploid. Am I correct?
Yes, you're correct so far.
Naomi Anderson.
Uh-huh.
With mitosis, like she said, the diploid cells are identical to each other and the parent cell. And then with meiosis, it's 4 cells and they're all different.
They're all different from the progenitor and from each other. Yeah, so that's stated very nicely. And that is the case. The goal of mitosis is to be exactly the same. The goal of meiosis is to never be the same, always generate variation. All right, the next question, what is a karyotype?
Patrick Dow.
Okay,
Those are visual display of chromosomes.
yep, you're right. Karyotype is a picture. And what might we see in a person's karyotype?
We're looking at their sex chromosomes and their autosomes.
Uh-huh. And if they had a genetic problem, what might we see? Or
Abnormality in the karyotype.
yeah, you might see an abnormality. You might see a chromosome with an odd shape, different from normal. Or someone else, take question 7. A trisomy is an extra copy. That's something else you might see in a karyotype, is an extra copy. And so for most chromosomes, what is the consequence of a trisomy? 3 copies instead of 2.
I can do this one. Abby Beck.
Uh-huh.
I think that the consequences could be like diseases within the child or like having a miscarriage or health problems.
Yeah. And so, for example, Down syndrome. That's question A. Down syndrome is the result of what chromosomal aberration?
It's the one we were just talking about, isn't it? The tri.
It's a trisomy of chromosome 21.
Yeah.
But now, so of the 23 chromosomes, haploid set of chromosomes, any one of them can exist as a trisomy. So what happens in the case of most trisomies, most chromosomes that are present in 3 copies? What happens to the fetus?
It causes a lack of development and
I think somebody's talking, but I can't hear it.
I said it causes a lack of development and
Can you turn up your mic? I'm not hearing whatever you're saying. Can someone else?
What? Michael here? Can you hear me?
Yeah.
Okay. I think she was saying that it could cause a lack of development and growth for the person, for the individual.
Yes, that's true. In fact, most trisomies are fatal. The fetus is never born because it aborts during development in the womb. Now, a few trisomies are actually result in live births, but most of those that do result in birth, the child lives a very short time, anywhere from a couple of weeks to maybe a few months. And it's the rare trisomy that an actual adult will result. Down syndrome is one of them, chromosome 21. And remember, there's also an aberration where an X chromosome is missing. That's always results in a female that develops into a normal person. I shouldn't say normal, into an adult, but the female functions are missing. So there's no puberty, and the woman is infertile. Okay, we've already discussed number 9, so let's go to number 10. This is a multi-answer question. I have a list of structures on the left and descriptions of those structures on the right, and the goal is to match them up. So, nucleolus, what does that match up with? Let me prompt your memory. Here, nucleolus. Here's a nucleolus. What does it do?
Compose RNA or RNA.
It, produces a type of RNA, ribosomal RNA. So it's number C here. Nucleolus produces ribosomes. The endoplasmic reticulum. From that list, what matches up?
Isn't that D, the site of collaboration of RNA, tRNA, mRNA for the protein synthesis?
Yeah, that's right. Endoplasmic reticulum is where all the different types of RNA come together to make a protein. Ribosomes. there's actually two answers there that are right. One of them is more right than the other. Think about what ribosomes do in the endoplasmic reticulum.
Does it create a peptide bond?
That's one of the possible answers. It's one that I don't prefer. There's one that I think is a little more accurate than that.
By the way, this Taylor.
Is it G assembles proteins in the endoplasmic reticulum?
Yeah, I think that's probably the most accurate description of what ribosomes do. But if your view, as Ben says, is to produce peptide bonds, yes, ribosomes do contribute to the production of peptide bonds. Okay, nuclear pore. Remember, everything is coded within the nucleus, and then some things end up in the cytoplasm. So think about what the function of the nuclear pore is.
Would it be H?
Yeah, it's an exit route for ribosomes or anything else that needs to get out of the nucleus and into the cytoplasm, like mRNA would also use a nuclear pore. Chromatin. Think about the relationship between chromatin and chromosomes.
Is it A? A long relaxed chromosome?
Okay, I know someone's speaking, but I cannot hear them. Can anyone else hear what she's saying?
I think she said A. Her name is Samaira.
Yeah, okay. A is right. Chromatin and chromosomes are really the same thing. It's just that when we talk about chromosomes, they're the condensed version that we can actually see in a microscope. Chromatin is what we see during interphase, or actually don't see because it's long, relaxed, indistinct chromosomes. But they're both constitute the genome of the organism. Cytoplasm. From the list.
Isn't that I? The watery cell interior containing organelles.
Yeah, that's what it is. If we look at this picture down here, which emphasizes the nucleus, but the cytoplasm is also on display there. So you can see the cytoplasm contains all of the organelles that are found in a cell. And finally, the nucleus. Anybody zero in on it? How does the nucleus relate to any of these things?
Would it be F?
Yeah. It's the organelle that contains chromosomes. So you're right, it is F. Okay. The question number 11, we've already gone over that, so we'll skip to the next one. What is the function of the centromere? And while you're thinking about that, I'll show you a picture. You see the centromere in this little diagram at the bottom of this slide. So what is the function of this little yellow body? We're going to talk about this in the next slide set. But let me just tell you that the centromere is where the microtubules attach to the chromosome. And this attachment is critical for the separation of chromatids and the movement of the chromosomes from the center of the cell to the edge. And we'll talk about that in a little while. How does cytokinesis differ between plant and animal cells? That should be an easy one. Who wants to say what it is? There it is. So what's the difference? On animal cells, what's the critical structure for cytokinesis? It's the contractile ring that pinches once the nucleus has divided into two nuclei and they've gone into opposite ends of the cell, then this ring pinches the cytoplasm into two so that you end up with two cells. Now that can't happen in plant cells because of the cell wall. So since you can't just pinch the cell into two cells, a new wall is formed in between the two budding cells. Vesicles containing cell wall components fuse, make a cell plate, and that ends up forming a wall between the two daughter cells. Okay, the haploid phase in humans is very short compared to the diploid phase, but there are some differences between what happens in males versus females in this haploid phase. So let's talk about just a couple of these differences. Anything come to your mind? Difference between males and females in the haploid phase.
Patrick Dowell here. Women are born with lots of eggs. Males are born without sperm yet.
Yeah, that's a big difference. Females are born with lots of products of meiosis. Males have none until they reach puberty. Another big difference, think about how long the reproductive phase is between males and females.
Women about done around 55 years of age. Men will continue to produce sperm their entire life.
They can, yes, and that is a big difference. The reproductive, the end of the reproductive phase in males is not defined, whereas in females it is. It starts with menopause. Okay, this number 15 is related to some of the previous questions. The result of a mitotic event is to double the number of cells, and they are identical to each other. Whereas in meiosis, it's very different. Instead of one cell generating two, one cell generates how many? In meiosis, you start with one cell and you end up with how many cells?
Was that 4?
Yes, there are 4 products of meiosis, of which how many of them are identical to the parent?
Zero.
0 is right. That's one of the main goals of meiosis, is to make sure that the progeny are not identical to the parent and not identical to each other. It's a source of variation. And that brings us to 16. Environmental conditions on planet Earth are continuously changing and organisms must adapt. To do this, the genetic makeup of every species must continuously change over time. What are the sources of genetic variation that allow species to evolve? We've talked about a couple of them already. If you think about meiosis and mitosis, the difference between them, what happens in meiosis that generates genetic variation? I don't think I have a picture of.
Isn't
I don't. Okay, go ahead.
isn't that when the chromosomes cross over?
Yes, that's one of them, crossing over, which is called a genetic recombination. And what else happens in meiosis? When the chromosomes go to the metaphase plate and they have to decide which way to go, how do they do that? That's actually question number 18. And they line up here in metaphase, and each pair of chromosomes is going to divide into 2, and one of them is going to go one way and one is going to go the other. What determines that?
Random selection.
Random. It's called random assortment or independent. So those are 2 big, very important sources of genetic variation. And we've also talked about others in addition to that. There are 2 others. A major one we're going to talk about later today, that's mutation. And the other is the random choice of fertilization. So when an egg is fertilized, the choice of sperm is totally random. So again, that's a source of variation. 4 different types of variation that organisms can rely on to help them over time adjust to changing environmental conditions. Number 17 is related to that. This is a true or false. Crossing over is the physical contact between chromosomes from opposite parents and the exchange of chromosome parts. Is that an accurate statement? Anybody want to guess? Yeah, there's no trick there. It is an accurate statement. Crossing over, which we just discussed, is when chromosomes in meiosis, this doesn't happen in mitosis, but in meiosis, they contact each other and exchange parts. And the next two questions we've already talked about, so we don't need to do it again. All right, that's the worksheet for today. Any questions you'd like to follow up on? All right, then I'm going to stop sharing that screen. Do you see illustration of experimental science?
Yes.
Yes.
Okay. When I showed this slide to my other class, I had trouble advancing the slide. So let's see if it works for you. All right, what I'd like to do now is describe to you a project that was done at Cornell University when I was on the faculty there by one of my graduate students. His name is Satoshi Inoue, and he did this project for his PhD thesis. Oh, why am I telling you about it? Well, because it's directly related to what we're discussing in Bio 130. It's directly related to cell division, in particular mitotic cell division. It's related to mutations, how you make targeted mutations, how we did it before the existence of CRISPR, and it deals with data analysis and how you interpret data. So the question that Satoshi asked was, what is the role of a motor protein called Dynein? What is the role in mitotic cell division? Now, I'm going to try to advance. Did I advance the slide? Can you see?
Yes.
And a picture of Satoshi?
Yes, sir.
You see that? Okay. So after doing this work, we published the results in a journal called the Journal of Cell Science. This is the title page, and you can see the authors. Satoshi's the senior author. I'm there at number 2. I was the principal advisor to Satoshi as he did the work. And here is the summary. The whole paper is available online if anybody ever wants to look at it. Not really necessary for you to do that for purposes of this course. So what was the experimental system that Satoshi was working on? It's a fungus called Fusarium solani, also known as Nectria hematococca, which is a plant pathogen. Now, this fungus can grow in an agar medium, which you see on the left. It looks like an ordinary mold. And if you look at a colony like that under the microscope, you'll see that it's composed of all these little threads called hyphae, which taken together are called a mycelium. So this entire colony here would be called a mycelium. And all of these cells can reproduce by mitosis, never or rarely by meiosis. So we have many, many mitotic cell divisions, which can give rise to spores. And because this is a haploid dominant life cycle, the spores are haploid and they're asexual. They're these long needle-like structures, and here you see them in 3D. And as I said, the fungus cause diseases of quite a variety of different plants. This happens to be soybeans, and you can see that the diseased ones are growing very poorly. In the field, they would never yield any soybeans. So it's a very serious pathogen of crop plants. Well, this is the organism that Satoshi used to look at mitosis. And the focus of his attention was on this protein called dynein. So what is dynein? It's a family of proteins called motor proteins. And why are they called motors? It's because they provide the force required for chromosomes to move. When mitosis occurs, chromosomes line up in the center of the cell, and then they move to the edges of the cell. They don't do that passively. They don't just float over. Something has to move them. And in this case, dynein actually pulls them. Dynein molecules travel along the microtubules in cells in what's described as a walk. And it's an active process because ATP is required. They have to expend energy in order to move. So the chemical energy in ATP is converted to mechanical work. And at the bottom here, it illustrates the process. And you can imagine this, if you take your two fingers and walk them across your desktop, that's the motion that dynein makes as it walks along a microtubule. So each one of these is a dimer, 2 molecules of dynein, a blue one and a yellow one. And it starts out, the blue one is on the left, and it moves over, so it's on the right. And then the yellow one moves from left to right, and so on. And for each step, ATP is expended so that energy is available for steps to be taken. Now, at the bottom here, this row of ovals is a microtubule. So this is how a motor protein works. And this works not only for moving chromosomes, but for example, when you flex your muscles, same thing happens. Dynein, in combination with another protein called actin, walks along microtubules to generate the force that your muscles exert. This is a diagrammatic illustration of where dynein is involved. So if you look on the upper left, you can see the chromosomes. This would be a metaphase plate. Chromosomes are lined up there. And you can see the green lines are connected to the centromeres of the chromosomes. And the other end of each microtubule is connected to a body called a centrosome, or a spindle pole body. And then beyond that, there are more microtubules which reach out to the edge of the cell and form a cluster called an aster. And what you can see here, the little walking figures are generating force in 2 directions, and the force is always toward the centrosome. So the dynein is moving the chromosome from the center toward the centrosome. And on the other side, the microtubules in the aster are moving the centrosome to the edge of the cell. So there are 2 forces, both focused on moving, getting chromosomes to the edge of the cell. And you've seen this picture several times before, and I show it just to help you relate what I'm talking about to what you already know. And that is that metaphase and then anaphase, where the chromosomes separate, The pink is the microtubules. You can't see them, but the little motor proteins, Dynein, are walking along the microtubules, pulling the chromosomes to the edge and out beyond where the chromosomes are. This is the area of the aster, which where the microtubules are connected between the centrosome and the edge of the cell, pulling everything out to the edge so that there's a nice, clear, and clean division between the chromosomes on one side of the cell and the chromosomes on the other side. And at the bottom here, just let me emphasize that the microtubules on which Dynein walks are connected to the chromosomes by the centromere. So when the two chromosomes have to go in opposite directions, Dynein pulls on one side to the right and on the other side to the left. So the question then was, what would happen if one of the motors was missing? And the strategy to address this question was disrupt the gene encoding Dynein. And this was back in the 1990s. That paper I showed you was published in 1998. So this was back long before CRISPR. And just to remind you, CRISPR is a gene targeting technology where you make a guide RNA, which is complementary to your target. Target is in the chromosome, and you want to mutate this target gene. And so you couple the guide RNA with this enzyme called Cas9, and when the target is fixed, Cas9 makes a double strand cut, and there's the mutation. Okay, as I said, this was not available in the 1990s, but yet we wanted to do a targeted mutation. We wanted to specifically target the Dynein gene. So how did we do that? We took advantage of homologous recombination. So this is the same process by which chromosomes exchange parts. We used the same process to specifically target the Dynein gene. So at the top here, the top bar, this is very complicated. Don't let it blow you away. I copied this out of Satoshi's paper and then tried to point out what's important here. So this top open bar is a chromosome. And on this chromosome, I colored here in red, is the Dynein gene. And we knew that because of the sequence. So we made a construct called a transformation vector in which we took the Dynein gene and inserted into it another piece of DNA which encodes a drug resistance gene, labeled here as HyG-B, resistance to the drug hygromycin B. So now we have a mutated gene on a vector. We rely on homologous recombination. Two recombination events replace the wild type gene with a mutated gene so that you end up with the chromosome on the bottom here. It's the same chromosome. You come along, you come to the Dynein gene, but instead of being intact, it's disrupted by another sequence, the hygromycin B sequence. And then at the end of that, the Dynein gene picks up again. So this gene is dead. It does not function at all, and we can show by other molecular techniques that that was true. So now that you've done this, made a mutation, how do you analyze it? Well, you can do it macroscopically, just look at the colonies, or you can do it microscopically by looking down a microscope. And Satoshi did both. So we have a macroscopic phenotype which we'll talk about here. And we'll talk about the microscopic one on the next slide. So you see these two red circles. The one on the left is a Petri dish with agar medium that has no drug. It's just a non-selective agar medium. And I should mention, going back to the disruption, most of the insertions are homologous. That is, the target gene gets disrupted. But occasionally, this vector will insert somewhere else in the genome. So it leaves a Dynein gene intact, but it still introduces the hygromycin B gene. So it still has a selectable marker, but it's Dynein plus. And so if you look at growth on a medium without drug, non-selective, wild type grows just fine. The control, this is the one where the vector inserted somewhere else, it grows just fine. Look at the Dynein mutant. And this brings up sort of an interesting side story here. Satoshi started this project with the goal of getting a Dynein mutant. And after some long period of time, he came to me one day and said, look, I'm not getting anything. It's not working. So I said, well, let's think about what you're doing. You're disrupting a gene for a motor protein. And I can think of two possibilities why you're not getting anything. One is that the mutation is lethal. So every time you mutate the gene, the fungus dies. If that's the case, you're never going to get a mutant and your project is over. You'll have to find another project and start again. The other possibility is that the Dynein mutant is just sick. And that is indeed what happened. So what Satoshi, excuse me, what he started to do, instead of growing up his plates for a week and then looking, he grew them for a month. And he would cut out all the colonies that were growing fast, just waiting to see if anything was growing slowly. And sure enough, after a month, he spotted these tiny little colonies, which are indeed Dynein mutant. So even though they grow very slow, they do grow enough for analysis. And if you look at the Petri dish on the right, this is exactly the same, except the medium has a drug in it, hygromycin B, a selective drug to which wild type is sensitive. So you see where the wild type is plated, there's no growth at all. Whereas the two mutants, the control mutant and the Dynein mutant, they're growing just as well on the drug medium as they do without the drug. That's because they both have the hygromycin B resistance gene. The control has an intact Dynein gene, whereas the mutant has a totally dead, defective Dynein gene. So this is what they look like to the naked eye. You just look at Petri dishes. And if you look down a microscope, you see the following number of things. So if you just look at the cells of the hyphae growing and compare wild type with mutant, wild type looks pretty healthy, and the hyphae are straight, whereas the Dynein mutant, the hyphae are twisted, gnarled, and don't look healthy at all. And if you remember, these are the hyphae you're looking at. This Dynein mutant is this tiny little thing that can just barely grow. And if you look at a microscope, you can see that it's really unhealthy. This stain called DAPI targets just nuclei. So if you look at the nuclei in wild type, there's one per cell. They're evenly distributed, normal shape. If you look at nuclei in the mutant, there are multiple nuclei per cell. And many of them are misshapen. So that you can see just from the physical distancing that the nuclei are in trouble. If you look at the spores that are produced, spores in the wild type look normal, whereas in wild type they're small and misshapen. Then one of the most dramatic things was movement of nuclei. As a cell grows, the nuclei or nucleus closest to the tip of the cell moves toward the tip. So, and that's what this graph shows, the distance from the nucleus to the tip. And the time scale here is 5 minutes. So in the wild type or in the control transformant, which has an intact Dynein gene, nuclear movement is very fast. In the course of 5 minutes, it moves 20 to 40 micrometers. Whereas if you look at movement of the nuclei and the Dynein mutant, they don't move at all. They start at 10 and they end at 10. So this is over a time course of 5 minutes. If you wait long enough, nuclei in the mutant do move, but much more slowly because they don't have the motors to move the nuclei. So this was Satoshi's project, and what can we conclude from his results? Well, first of all, the Dynein motor protein is required for normal mitotic cell division and for growth. You can see just from looking at the colonies that growth is very highly impacted. Now, what's the significance of that? Well, it contributes to our understanding of the molecular basis of mitosis. And now we know that this Dynein motor is required to move chromosomes to the poles of the cell during mitosis. Without Dynein, cell division is very slow, and as a result, growth of the fungus is very slow. How is this related to Bio 130, what we're talking about? Well, we spent a lot of time talking about mitosis and mitotic cell division, and now we know that one element of that we hadn't talked about before is, in fact, a motor protein called Dynein. And this functions in both anaphase and telophase of mitosis. It's related because we've spent a lot of time talking about mutations and how mutations occur and how you can make targeted mutations. And the choice technology nowadays is CRISPR, but before the days of CRISPR, we could still do it by taking advantage of homologous recombination. And finally, this is an illustration for you to consider of how real-world scientific investigation is done. So I think that's the end of that. Yes. Any questions about this project, how it was done, why it was done, what it means, why I went to the trouble of describing it to you? Did it make sense? Anybody confused about any part of it? Are there gaps in my explanation? Okay, you're pretty quiet, but if there is no discussion of this, I will Do you see Mendelian genetics?
Yes.
Okay. This is the next topic for the course. And we're going to spend considerable amount of time on this, and you're going to have a worksheet on it. Mendelian genetics arose during the mid-1800s, which was an era of great ideas in many places in the world. Two of the main actors, at least as far as biology is concerned, are Gregor Mendel and Charles Darwin, but at the same time, Karl Marx was coming up with economic theories that shook the world and continue to do so to this day. And during that same era in the United States, there was a civil war, which was fought to determine if the country was going to, if it was going to be one country or two. And Lincoln was almost single-handedly made the effort to make sure that the country ended up as one country, not two. But let's first talk about Mendel and Darwin. Gregor Mendel was a monk. He lived in a monastery in Austria. He worked with peas. He knew nothing about molecular biology or DNA or chromosomes or genes. He was just interested in inheritance. How are traits inherited? And he published the results of his work in the mid-1800s. Charles Darwin, we're going to spend a lot of time talking more about him. He published in 1859 a seminal book called The Origin of Species, and from that arose the theory of evolution. And along with that, the concept of natural selection and how it shapes speciation as environmental conditions change. As far as we know, these two had no knowledge of each other. They did their work independently, but there is a relationship between the two bodies of work. I want you to remember this. I have it circled in red. Darwin established that natural selection is the force that controls evolution, that controls species change. Mendel provided the rules for Darwin's observations, the rules for the basis of variation that underlie natural selection. So the two go hand in hand, and between the two, it caused a revolution. Okay, prior to Mendel, what did geneticists do and what were their results? Well, people who were interested in inheritance would take two parents, parent one and parent two, and cross them, and then look to see what progeny they got. And they'd almost invariably get something like you see here in this graph, a bell-shaped curve, where the type of progeny are plotted against frequency. So most of the progeny would have characteristics of both parents. That's the majority. A few of them would look a lot more like one parent than the other, and a few would look more like the other parent. But you couldn't make sense of this in terms of what's causing inheritance. So this went on literally for centuries with no progress. Mendel did two things that led to clarification, listed here on the left. He did inbreeding and he did F2 analysis. Now, what do those two things mean? It's critical for your understanding, and we'll talk about that next. First, definitions. We have to know the language before we can discuss. What is a cross? It's the breeding of two individuals, either plants or animals. P1 is crossed with P2. And that is the parental generation, which leads to what's called an F1, the first filial generation. And what is the F2? It's a cross of two F1 individuals. So you take the progeny of the first cross these progeny to each other, and you get an F2. And this is half of Mendel's genius. He actually did this second step, the F2. Phenotype and genotype. Phenotype is what you see. So you can describe the phenotype in words: tall, short, black, pink. A genotype is a symbol. So it's a representation of the actual genetic makeup of an organism. And so you choose a symbol. In this case, we choose T to represent tall and make it a capital because it's dominant. And we choose a lowercase t to represent the alternative trait, which is short. And because short does not occur in the presence of tall, we call short a recessive. So how did Mendel actually do his experiments? Well, it turns out that peas are an inbred, and that caused a great deal of problem for Mendel. But let's consider what an inbred really is. It's a mating of closely related individuals. And so if you keep doing that, if you keep taking progeny of two closely related individuals, cross them to each other, and keep doing that for successive generations, you finally end up breeding out all the heterozygosity. So that all the loci that are left among your final progeny, they're all homozygous at all loci. And this is called a true breeding line. So at all loci, they're either dominant alleles, I'll talk about alleles in a minute, or recessive alleles. In contrast, a hybrid is the mating of distantly related individuals. Most of the loci in a hybrid are heterozygous. Now, essential to Mendel's success, he chose P as a subject, and he chose 7 different characteristics, which we'll talk about in a minute. And before he started doing any crosses, he did a series of inbreeding generations. This slide is a restatement of what I just said. What is an inbred and what is inbreeding? You achieve homozygosity by inbreeding. And here are the 7 traits that Mendel chose to study. He looked at seed shape, and I should emphasize here that all of these pictures represent results after inbreeding. So Mendel took true breeding lines, one with round seeds and a true breeding line with wrinkled seeds, and crossed them and looked at the F1, and they were all round. Same thing with seed color, yellow and green, the F1, they're all yellow. With flower color, purple and white, the F1 is all purple. With pod shape, constricted and smooth, turns out smooth shows up in the F1. Pod color, yellow versus green, it's green in the F1. And flower position, if flowers can either be attached to the stem or to the terminal bud. And it turns out that the ones attached to the stem show up in the F1. And as far as plant height is concerned, it's tall that shows up in the F1. So Mendel went to the trouble of making true breeding inbred lines of both round and wrinkled, yellow and green, purple and white, and so on. And he did this in a very laborious way because he had to. Peas are naturally self-pollinating. That means that before the flower even opens, pollen is shed and fertilizes the stigma, the female part. So to prevent self-pollinating, he had to open the flower bud before it was mature and take a forceps and remove the stamens, remove the pollen, which is the male gamete. And then close the flower back up again. And if he wanted to make a cross, he would go to a different plant, take the pollen from that plant and put it on the first one. And all of this had to be done very laboriously with forceps and tiny little paintbrushes. And so I'll describe this Mendel's first experiment, and then we have to stop for the day because we're out of time. But this is key step 2. Key step 1 is to develop inbreds, true breeding lines. Step 2 is to continue to the F2 generation. So the parental generation, and these are all phenotypes, the true breeding parents for the parental generation, purple was crossed with white. And in the F1 generation, instead of getting a continuously spreading population of progeny, he got just one type, all purple. And then he took 2 of these purple plants, purple progeny, and crossed them with each other. And what a surprise. Instead of all coming out purple, some of them were white. And when he counted, it turned out that for every white flower, there were 3 purple ones, a ratio of 3 to 1. And so these are the phenotypes. And let me just quickly go through the genotypes of a monohybrid cross so that you can think about what actually happens here. If you take the parental plants, we know that they're true breeding, which means that if we say that purple is dominant and we represent that with a big P, then one parent is big P, big P. And the other parent, by definition, is little p, little p. We say this because they've been inbred. They're true breeding lines. So all the gametes made by the purple plants, they're all big P. All the gametes made by the white ones are little p. And so now the F1 generation is all big P, little p. And so they're all heterozygous, but they're all purple because big P, purple, is dominant. But now the gametes made by these heterozygotes, half of them are big P and half are little p. And so from this, you construct what's called a Punnett square. And you're going to be doing a lot of these as you work out problems on the worksheet. But you plot the eggs against the sperm. So the eggs can be either big P or little p. Sperm can be either big P or little p. So if a big P sperm fertilizes a big P egg, you get big P, purple. If a little p sperm fertilizes a big P egg, again, you get purple, but it's a heterozygote, big P, little p. And there's another one that will form here. It's only if a little p sperm fertilizes a little p egg that you get little p, that's recessive and that is white. So the phenotype here is 3 purple to 1 white. The genotype is 1 big P, 2 big P, and 1 little p. And I have to stop at this point because we're out of time, and we'll pick it up here on Wednesday and continue on talking about alleles and dominance and recessiveness. So are there any questions quickly before we go?
I just want to make sure it is right. So we have one worksheet that due on Wednesday, right?
Yes,
And the worksheet that due today would not be graded, right? So it doesn't matter if you submit it or not submit it.
that's right. It was for purposes of discussion only. The worksheet that's due on Wednesday, I will grade. That's the one that has the overlapping circles. And it's on mitosis and meiosis.
Thank you.
Okay, anything else?